Welcome back, everyone, to Vox Markets. My name is Paul Hill, and by popular demand, I'm delighted again to be able to speak to CEO Alastair Smith and Chief Development Officer Neil Bell of Avacta, a leading therapeutics and diagnostics firm based on its two platforms, the Precision and Affimer technology. So welcome, gents.
Hi, Paul.
Hi, Paul. Yeah. Well, big congrats on last week's major news of starting first patient dosing of Doxorubicin in phase 1 clinical trials at the Royal Marsden. But before sort of digging into the details, Alastair, I don't suppose you could just quickly remind investors just broadly how Doxorubicin works and also how it fits in the grand scheme of things of the Precision platform.
Yeah, sure. Yeah, so the precision chemistry is designed to prevent the chemotherapy like Doxorubicin from entering non-tumor cells and therefore prevent them from harming that healthy tissue such as heart tissue. So in that state where the chemotherapy has been modified with the precision technology, we call it a prodrug. And the prodrug is a compound that's inert when it's administered, and then it's metabolized. So the precision chemistry is removed by an enzyme in the body, and that then releases the active drug. And so the precision chemistry is removed by an enzyme called FAP alpha, and that releases the chemotherapy so that it can get into the local cells and leads to cell death. And the FAP alpha enzyme is present in high concentration in the tumor microenvironment and only at much lower levels in healthy tissues. So you get that release and that activation, so that release of the active chemotherapy predominantly in the tumor when it encounters FAP alpha and therefore spares the healthy tissues from that same level of exposure to the active drug. And that's intended to improve safety, therapeutic index, and thereby efficacy and better outcomes for patients.
And why was Doxorubicin sort of selected as the lead chemotherapy for this prodrug sort of like testing and concept?
Yeah, so it's a good question. So anthracyclines, of which Doxorubicin is one, that they're the backbone of cancer chemotherapy. Doxorubicin's been around many years. It's widely used in cancer care. It's a generic drug. By the nature of its mechanism of action, Doxorubicin's got significant off-target side effects, including cumulative cardiotoxicity, and therefore patients are limited to quite a small number of cycles, up to 6 cycles, regardless really of the clinical efficacy. So a safer form of Doxorubicin could allow longer treatment cycles, may also be suitable for older people who are ineligible for Doxorubicin because of the cardiotoxicity. And on the commercial side, Doxorubicin's $1 billion generic market. So we believe that a proprietary, safer, and potentially more efficacious form of Doxorubicin could have a market size well in excess of $1 billion annually.
And then the efficacy of Doxorubicin, if you could sort of like just park the safety problem it has with it, but if you just look at the sort of how effective it is for sort of like helping cancer patients, how powerful, how effective is it? Is it mainly the safety issues rather than the efficacy?
Yeah, I mean, I'll comment. I'll let Neil add something. So generally speaking, chemotherapies are quite effective cancer treatments, but it is the tolerability for patients that ultimately limits their use, I'd say, Neil.
Yeah, so Doxorubicin is actually an old drug, but it's actually a very effective drug. And so it does kill tumor cells, but unfortunately it's got off-target, what we call off-target toxicity. And that's where the damage to the heart comes in, damage to the bone marrow and other organs and tissues. So with AVA 6000, we're able to limit that damage, hopefully, in the human situation, and that's where the preclinical studies have come into play as well.
Yeah, and just on that one then, Neil, how did the prodrug, Doxorubicin, perform in preclinical studies?
So we've got quite an extensive non-clinical, as we call it, preclinical package actually, and it's been shown that cell permeability, the DNA binding, and the associated cytotoxicity of AVA6000 is actually significantly reduced compared to conventional doxorubicin in what we call in vitro models, but also in animal models as well, in mice, rats, and dogs, which are the typical models that you'd expect to actually perform from the regulatory setting.
Yeah.
What we expect AVA6000 to have basically is an improved therapeutic index relative, obviously, to conventional doxorubicin, increased concentration of doxorubicin in the tumor as well, and hopefully that the patients, through this more precise targeting of the tumor, will actually tolerate either higher doses or an increased number of cycles of AVA6000, relative to conventional doxorubicin.
And what types of cancer would it best be suited for the treatment? Is it sort of like, I mean, it's obviously tumours, it's solid tumours, but what type is it? Sort of like ovarian, pancreatic, I don't know, lung cancer, or does it actually solve most of them because they have this enzyme that effectively will be activated? Effectively, the fat enzyme will be activated, and then the chemo payload will then be deposited on the tumour. Is it any specific sort of like types of cancer that would be help really sort of like beneficial?
Well, yes, you're right, Paul, that there are two main types of cancer. One is solid tumours and the other are blood cancers, as we call them. We have done all of our preclinical work on solid tumour cancers. So we expect AVA6000 to respond to solid tumours. And these solid tumours actually have fibroblasts on their within the tumor microenvironment, the stroma, and those fibroblasts actually express the enzyme, the FAP enzyme, which then converts AVA6000 into active doxorubicin.
Yeah.
So in terms of different cancer types, it's pretty well known in the literature that there are a number of cancer types which actually have high levels of FAP. And we're actually recruiting all of those different cancer types into our study, into our phase 1 study. And then we'll start to, for our dose expansion part of the study, we'll start to actually select the different type of, 1 or 2 or even 3 types of tumor, which will be more responsive, let's say, to AVA6000.
Yeah. And then just as it's a bit technical, I guess, but as the sort of like the tumor grows and it starts to spread and becomes metastatic around the body, there is a sort of like a field of knowledge saying that you get these circulating tumor cells that effectively is that tumor which is shedding into the blood and hence allows it to spread around the body. Would that potentially, cause an issue in terms of leakage of the FAP enzyme, or is this it's so rare that it happens that it isn't going to make a massive difference in terms of toxicity?
Yeah, in fact, circulating tumor cells are incredibly rare, and so we consider this to be a risk. And I guess you're really asking about how AVA6000 will perform when it's infused into humans. Doxorubicin, conventional doxorubicin, when it's infused into humans, into patients, actually disperses very quickly into the tissues. Whereas AVA6000, we anticipate it will actually not disperse as quickly. It will actually circulate around the body until it actually finds the tumor site. And that's where the 6000 or doxorubicin will be separated away from the substrate by the FAP enzyme. That's where we expect to find the highest concentration of the FAP enzyme.
Yeah, okay. So effectively, it's with a bit of luck through the clinic, and that's why we're doing the clinical trials, but it should be, in theory, given I think the CTCs are sort of 1 in a million, it should be de minimis and hardly be noticeable. How is, just broadly, Alastair, how is the sort of the trial being sort of like structured in terms of phase 1 and phase 2, or stage 1 and stage 2?
Yep. Well, we would call them phase 1A and phase 1B. So
okay, sorry.
That's fine. So the dose escalation, dose expansion phases. Yep. So the dose escalation phase is where the dose is increased in steps. I'll let Neil add some more detail in a moment. And that is in small cohorts of patients to identify the maximum tolerated dose. And then the dose expansion phase Neil alluded to a moment ago, where having brought in a wide range of cancer types into that dose escalation phase, we can narrow down in the dose expansion phase to 1 or 2 or possibly 3. You got anything to add to that, Neil?
Yeah, no, it's exactly, we've actually designed this study with experts, and it's been through the regulatory authorities as well. So the study is actually designed initially, it's split into 2 parts, as Alastair said, the dose escalation part, which is actually designed to really understand the safety and tolerability of escalating doses of AVA6000. But also designed to understand how AVA6000 works in the body. And then we can also select an appropriate dose, which we might call the maximum tolerated dose, or it may actually be the recommended phase 2 dose. And we will select a dose, which will then go into the dose expansion part of the study.
Yeah.
And that's where we'll select 2 or 3 cohorts of patients who have the same tumor type. Could be ovarian, it could be pancreatic, it could be lung, and we'll actually start to understand and confirm the selected dose, whether it actually it continues to be safe and well tolerated, and in addition to that, it'll actually provide us with some early efficacy signals as well.
Right. And just on your selection of actual patients, how specific are you being in terms of, is it got to be essentially sort of late stage sort of tumor patients, in terms of those relevant types of cancer, or is there any type of patient you definitely don't want, or how selective are you being with in terms of your patient recruitment?
Well, we're being very selective. That's all governed by our study protocol. And we're actually selecting a number of tumor types for our dose escalation part of the study. And those patients generally have been through all the normal treatment regimens that are required for their cancer type, and probably are at the end of the road in that respect. So we're dealing with patients who are actually being altruistic, and they're helping us to understand how AVA6000 works, and we hope it actually gives them some benefit as well.
Yeah. And is it like any impact at all from COVID, lingering COVID, in terms of recruitment of patients you could potentially use, or is that now, given with most people have been luckily double jabbed, the recruitment of patients should be fairly straightforward over the next sort of a year or 2 years?
It's been limited, I think, Neil.
Yeah, we've obviously hospital sites, if we were doing this on hospital wards, we would see a greater impact on the clinical study. But because we're doing it in dedicated phase 1 units, which are actually part of the hospital, the phase 1 units themselves have been working, albeit at a lower rate. So we are working with the premier UK oncology phase 1 sites, and we have done a lot of work with them, risk assessment and so on. We've actually prepared the sites very well. We have our own monitors that go in and actually check all the data. And at the moment, a lot of that has been done remotely. So we're working together in partnership with these hospital sites, and we don't anticipate that COVID will impact negatively.
Great. Okay. So assuming everything goes to plan, then what are sort of like your first initial sort of results you're hoping to get, and what are the sort of the endpoint goals of the study?
So the endpoint goal is that for the dose escalation part of the study, that we can actually confirm that AVA6000 is indeed safe to give to patients, and actually has a reasonable tolerability profile, and that we can also establish what we call the pharmacokinetic profiles of AVA6000 and Doxorubicin in both plasma and in urine, which will give an early indication as to whether AVA6000 is being cut away, or rather the AVA6000 is actually being metabolized into Doxorubicin and the substrate.
Okay.
We're trying, obviously, to find the right dose to select for further evaluation. So if we can actually identify that dose in the dose escalation portion of the study, and then we can move into the dose expansion with the correct dose and the correct dosing regimen, then this will all be good news for us. So those are the points that we're really looking for. And obviously, if we see, early efficacy signals, particularly in the dose expansion part of the study, we're very excited about this. So we've actually designed the study to be very data rich, and we're looking forward to actually, reviewing and analyzing that data.
And what's the standard of care? What's the comparator you're doing? I presume is it Doxorubicin or is it a placebo?
No, we're actually allowed in this case, and this is all ethically controlled as well. So because these patients are at a very late stage in their disease, we're allowed to do an open label study, as we call it, so it's not double blind. And we're also allowed to do a single arm, which is just AVA 6000 alone. As we progress into phase 2 and phase 3 studies, then you'd expect those comparisons to be designed into the study.
Okay. And then just sort of looking through the crystal ball then, for potential, if everything goes to plan and we sort of like get the green light after the end of phase 1, which was that's sort of mid 2022, 23, is it, or something? Is that
It's mid 2023 for the conclusion of the phase 1 study. Yes.
So Alastair, if we did move into phase 2 after phase 1, what would that mean, and what sort of optionality would the group have? Would it fund it itself? Would it license the product out, or what sort of options would be available? What sort of timescales? Again, if we further through ball to bring the whole thing to market.
Yeah, so we are very committed to building a clinical pipeline. So taking drugs through the clinical development as far as we can, as is practical. To the point where we add sufficient value that we can realise that for shareholders through a license deal. So, as Neil indicated, the clinical development group that Neil's established is more than capable of taking drugs through phase 1, phase 2, and even into phase 3 if we really wanted to and decided to. So, from our perspective, the commercial picture, if you like, is building a strong clinical pipeline, progressing our own assets through the clinic, quite extensively, as well as building a preclinical pipeline behind that, and licensing those assets at the appropriate time.
Yeah, okay. And just in terms of other big pharma, have they ever tried sort of this pro-drug approach for sort of doxorubicin at all? And if so, how successful has it been?
Well, more generally, the pro-drug approach, of course, has been addressed by other biotechs and pharma companies using FAP alpha to release the active chemotherapy or warhead is an obvious enzyme to choose because, as we said, it's highly upregulated in most solid tumors. There are some other enzymes. The main issue for all of the attempts that have been made to date, as far as I'm aware, certainly, is getting the specificity around FAP alpha. So just to expand on that very briefly, FAP alpha is a member of a family of proteins, of enzymes that are quite similar in what they do. So many of those are also systemic, i.e. they're spread around the body and not just specific to the tumor. So unless you can get your technology to be absolutely specific to FAP alpha so that only in the tumor microenvironment is the drug released, then you're going to get systemic release of the drug, and that gives you the same side effects and tolerability issues. So the thing that's unique, as far as I'm aware, about the precision technology is that we do have, Bill Bachovchin, the chemist at Tufts that we work very closely with and who invented the precision chemistry, has achieved a very high level of specificity to FAP alpha. So we get exquisite targeting in preclinical models and expect or hope to see that replicated in the human phase 1.
So what you're saying is that the previous pro drugs essentially have released the chemo warhead really outside of the actual tumour, whereas because the specificity coming out of the Tufts University and the precision platform is so much better and sharper, it means the toxicity should be a lot less in the healthy cells around the body.
Exactly. So getting that specificity to the tumour microenvironment is where previous attempts have failed.
Yeah. Okay, good. And then just broadly, Alastair, just sort of like news flow. I know you've got your interims in September. How do you sort of see, obviously, updating investors in terms of, obviously, the precision platform and, obviously, more broadly, the rest of the group?
Yeah, well, absolutely. Just as you just described, Paul, we've got our interims towards the back end of September. We haven't announced a date yet, but back end of September. And then we will give a full update across the business, as you just described.
Yeah, well, I'm looking forward to chatting then, hopefully in about a month's time. And I would just point investors just to have a look at there's a freely available research report by Trinity Delta, which puts a sort of sum of the parts on Avacta of roughly around about 270, 280p, and splits it down. And I think it's got a value for the products business, the Affimer and the precision technology of about 220, and the diagnostics roughly just 50p or slightly higher than that. So it's a good explanation of Doxorubicin, the precision platform, and also everything else. So there's plenty of shots on goal for investors. So look forward in hearing how the progress moves in due course.
Yep, look forward to speaking to you, Paul.
Thanks, guys.
Thanks very much.
Thanks, Paul.
Bye.
Thank you.
Bye-bye.